Heat map conveniently provide the hierarchical clustering of the genes and the arrays. And apart from this genes and arrays clustering, it also provides the information regarding the expression patterns of the genes. And this genes, arrays and expression patterns, they are all we can see in one visualization. To see all these in one visualization, first we have to cluster the genes and arrays. And for this purpose, we have to use the pairwise distances between the genes and between the arrays. To obtain these distances, we can use different methods that are available. And each method has its own drawbacks and positive points. The methods that are available for distances measurement, and two of them, they are very famous. One is the Euclidean and the Pearson correlation methods. And we cannot say with surety that which method we will use and we will get the best results. However, the choice that we use either of these two methods, it can affect the results. So we can use the library A map that is present in the bioconductor. And by using this library, we can obtain the data by using the command HC cluster and then parenthesis. If we are using the method for the calculation of the distance, and the method is Pearson correlation, then we have to use the argument within the command as Pearson correlation. And on the other hand, if we are using the other distance method, that is Euclidean distance method, then we have to use the argument that is the Euclidean distance in the command. So the command heat map, we can use this and we can get the heat map clustering and the expression data in one visualization. And for this purpose, we need the specifies the data set, which should be a matrix, and then we convert this normalized data to a matrix, and the arguments row V and column V take the clustering results that were generated earlier. The clustering results need first to be converted to dendrograms with the command AS dendrogram parenthesis. The last argument column specifies the coloring scheme to be used while generating the image. So by using these commands, we can get the heat map that we will see in the next slide. So this is the heat map representation that we will get after using the distance programs, either the Euclidean or the Pearson, and the resulting heat map resembles this diagram we are seeing. And in this, the hierarchical clustering of genes and the arrays are on the left side. There is a hierarchical clustering of the genes and the arrays, and on the top of the colored area respectively. In the colored area, we can see in the middle, every gene is represented by a color bar. So we can see a small red bar, green, red bar. So we can see different green and red bars. So color represents the down regulation green. So it means if the gene that we are representing here, one gene here, is a green bar, so it means that its expression is down regulated. Down regulated means that it has the less expression as compared to the control. Or the red regulation of the genes. And if the gene, some gene has the red bar, it means that its expression is up regulated in comparison with the control. So we can make a sense very easily by seeing this heat map, that some genes they are down regulated and some genes they are up regulated, and this comparison is in comparison with the control.